Picture this. You're standing on a beach at dawn, watching thousands of tiny loggerhead turtle hatchlings emerge from the sand. They've never seen the ocean before. They've never seen anything before. Yet somehow, these 2-inch-long creatures know exactly which direction to crawl. They reach the water and swim away from shore, beginning a journey that will take them across an entire ocean basin. Years later, after swimming thousands upon thousands of miles through a seemingly featureless sea, those same turtles will return to this exact beach. Not just the same coastline or the same country, but often the very same stretch of sand where they were born. How? Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. Tonight, we're embarking on a journey through one of nature's most extraordinary mysteries, animal navigation. We'll discover how creatures great and small find their way across deserts, through forests, beneath the waves, and through the air. We'll learn about invisible compasses, chemical maps, sound pictures, and mathematical calculations performed by brains smaller than a grain of rice. Some of what you hear may challenge what you thought was possible. Some may feel like magic, though it's all grounded in biology, physics, and evolution. This is the story of how animals navigate Earth, the ancient challenge of finding your way. For as long as animals have moved across the planet, navigation has been essential for survival. Think about what this means for a moment. A bird must find food sources scattered across a landscape, then return to its nest to feed its young. A bee needs to discover flowers miles from the hive, gather nectar, and fly back home. A whale must locate feeding grounds thousands of miles away, then swim to specific breeding areas. A desert ant, foraging for food in a landscape with no landmarks, must somehow calculate its way back to a nest entrance just millimeters wide. Get the navigation wrong, and the consequences are immediate and often fatal. You waste energy searching for home. You miss breeding opportunities. You starve, or your offspring starve. Natural selection has been fine-tuning animal navigation systems for hundreds of millions of years, creating solutions so elegant and sophisticated that scientists are still working to fully understand them. What makes animal navigation particularly fascinating is its diversity. There's no single solution that works for every species. A bat hunting in total darkness faces completely different challenges than a monarch butterfly migrating south. A salmon swimming up a river toward its spawning grounds navigates a very different world than a sea turtle crossing the Pacific Ocean. Evolution has equipped each species with the sensory tools and neural processing abilities suited to its particular ecological niche. But here's what's truly remarkable. Many animals don't rely on just one navigation method. They've evolved to use multiple systems simultaneously, integrating information from different sources to create a rich, multidimensional understanding of where they are and where they need to go. These creatures are using cues we humans can barely perceive, some that we can't detect at all without specialized equipment. As we drift through tonight's exploration, we'll encounter magnetic maps that work like GPS, celestial compasses that read the stars, chemical trails invisible to human noses, sound waves that paint pictures in the dark, and internal calculators that track every step of a journey. We'll see how tiny insects accomplish feats of navigation that would impress any human explorer, and how massive whales traverse entire ocean basins with pinpoint accuracy. So, settle in, relax, and let's begin our journey into the hidden world of animal navigation. We'll start with something humans can't directly sense at all, yet many animals use as their primary navigation tool. We'll start with Earth's magnetic field. Earth's magnetic field, nature's invisible GPS. Close your eyes for a moment and imagine that you could see Earth's magnetic field. Picture invisible lines flowing from the South Pole to the North Pole, arcing through the atmosphere and wrapping around our planet like the longitude lines on a globe. Now, imagine that these lines aren't just directional markers. They also have intensity, getting stronger as you approach the poles and weaker near the equator. And they have inclination, the angle at which they intersect the Earth's surface, which varies predictably with latitude. This creates something extraordinary. Every location on Earth has a unique magnetic signature defined by the combination of its field intensity, inclination, and direction. It's like an invisible coordinate system overlaid on the entire planet. Many animals can sense this field. They're equipped with biological magnetoreceptors, specialized structures that detect magnetic information and translate it into neural signals the brain can interpret. This ability, called magnetoreception, has been documented in an astonishing array of species: birds, sea turtles, salmon, sharks, lobsters, honeybees, ants, butterflies, newts, and even some mammals like bats and mole rats. Let's start with birds, where much of the groundbreaking research into magnetoreception has taken place. In the early 1970s, researchers Wolfgang and Roswitha Wilczko made a discovery that would reshape our understanding of bird migration. They demonstrated that European robins could detect the direction and inclination of Earth's magnetic field. This was met with considerable skepticism at first. Many scientists found it hard to believe that birds possessed a sensory ability so alien to human experience. But experiment after experiment confirmed it. When researchers placed migratory birds in special chambers where they could manipulate the magnetic field, the birds would orient themselves according to the magnetic direction, not the actual geographic direction. Rotate the magnetic field 90 degrees, and the birds would rotate their orientation by 90 degrees. It was direct, reproducible evidence that birds were using magnetism to navigate. But how do they detect it? Two main mechanisms have been identified, and birds may use both. The first involves tiny crystals of magnetite, an iron-rich mineral found in specific cells in birds' upper beaks. These magnetite crystals are physically rotated by Earth's magnetic field, much like how iron filings align when you place a magnet near them. This rotation can be detected by mechanosensory cells and transmitted to the brain via the trigeminal nerve. The second mechanism is even more remarkable. It involves a protein called cryptochrome, found in the retina of birds' eyes. When light hits cryptochrome molecules, it creates pairs of molecules with unpaired electrons, called radical pairs. These radical pairs are extraordinarily sensitive to magnetic fields. Even Earth's weak magnetic field can influence the chemical reactions these radical pairs undergo. This means birds may literally see the magnetic field as patterns of light and dark superimposed on their visual world. This quantum effect, this interaction between light, chemistry, and magnetism happening in individual molecules in a bird's eye, provides compass information. It's one of the few confirmed examples of quantum mechanics playing a direct role in a biological process that we can observe at the level of animal behavior. What's particularly clever about having two different magnetoreceptive systems is redundancy and specialization. The magnetite-based system in the beak appears to provide information about magnetic intensity, helping birds determine their position on a map. The cryptochrome-based system in the eyes provides compass information, helping birds maintain a heading. Together, these systems give birds both a sense of where they are and which direction to fly. The evidence for birds using magnetic maps has become increasingly compelling over the past 2 decades. Researchers found that juvenile birds can detect their position based on magnetic coordinates, even though they've never traveled before. They're born with an inherited magnetic map. When hatchling loggerhead sea turtles, which we'll return to later, are exposed to magnetic fields that exist at different locations along their migratory route, they swim in directions appropriate for those specific locations, demonstrating that even very young animals can interpret positional information from the magnetic field. Research on sea turtles has provided some of the most elegant demonstrations of magnetic mapping. Kenneth and Catherine Lohman, researchers at the University of North Carolina, conducted experiments showing that loggerhead turtles can detect remarkably subtle differences in magnetic field, intensity, and inclination. When placed in a pool surrounded by magnetic coils that reproduced the exact magnetic conditions found at different points in the Atlantic Ocean, turtles swam in directions that would keep them in the area where they typically develop and feed as juveniles. The turtles were essentially using a bi-coordinate magnetic map, but magnetoreception isn't limited to long-distance migrants. Even animals with more modest ranges use magnetic information. Honeybees use Earth's magnetic field for navigation, both near their hives and during longer foraging trips. They can integrate magnetic information with their sun compass and their memory of visual landmarks. Desert ants of the genus Cataglyphis, which we'll discuss in detail later, have been shown to use magnetic cues as one component of their navigation toolkit, though they rely more heavily on other systems. What's especially interesting is how different animals have evolved to use magnetic information in different ways, adapted to their specific needs. Some species use it primarily as a compass, giving them directional information. Others use it as a map, giving them positional information. Many sophisticated navigators, like migratory songbirds and sea turtles, use it as both. Recent research has also revealed some of the challenges that animals face when using magnetic navigation in the modern world. Magnetic storms caused by solar activity can temporarily disrupt Earth's magnetic field, and studies have shown that these disruptions can lead to increased vagrancy in migratory birds. The birds become disoriented when their primary navigation system feeds them faulty information. Even more concerning is the impact of human-created magnetic anomalies. Underwater electrical cables, iron-reinforced seawalls, coastal buildings with metal frameworks, oil drilling platforms. All of these can create local distortions in the magnetic field. For an animal relying on the precise pattern of Earth's field to determine its position, these anomalies could be like suddenly encountering wrong road signs on a highway. Some researchers worry that the proliferation of human-generated magnetic noise may interfere with the navigational abilities of marine species, particularly those that navigate near coastlines. The magnetic sense also has some limitations. It works best at certain times and under certain conditions. For birds using the cryptochrome-based system, daylight is essential. Many migratory songbirds begin their flights at dusk, likely because this is when they can calibrate their magnetic compass against the setting sun, using polarized light patterns in the sky. Once airborne in darkness, they maintain their heading using their magnetic sense. Research has also shown that magnetic navigation requires learning and calibration. Young birds go through a sensitive period where they learn to associate magnetic information with other cues like the position of stars or the sun's movement. This calibration process allows them to maintain accuracy as they grow and as they travel to locations with different magnetic properties. Salmon provide another fascinating example of magnetic navigation. Like sea turtles, salmon are believed to imprint on the magnetic signature of their natal river when they're juveniles. After spending years in the ocean, sometimes traveling thousands of kilometers, they use this imprinted magnetic memory to help guide them back to the same river system where they were born. Studies have shown that exposing salmon to magnetic pulses can disrupt their ability to orient correctly, providing strong evidence for magnetic involvement in their homing behavior. Sharks and rays, which inhabit an electrically conductive medium, may use a different mechanism for magnetoreception. As they swim through Earth's magnetic field, they induce weak electrical currents in the surrounding seawater. Their incredibly sensitive electroreceptors, called ampullae of Lorenzini, can detect these induced currents. By sensing the voltage differences created as they move through the field, they can infer their heading relative to the magnetic field. This is called electromagnetic induction, and it essentially allows them to use their electric sense to derive magnetic information. The sophistication of animal magnetoreception continues to impress researchers. It's a navigation system that requires no batteries, needs no calibration beyond what the animal does naturally during development, works day or night, and functions regardless of weather conditions. It's always on, always available, providing a stable reference frame for orientation and navigation. As we'll see throughout this exploration, magnetoreception often works in concert with other navigation systems. Few animals rely solely on magnetic cues. Instead, the magnetic sense forms one component of a multisensory navigation strategy, integrated with visual cues, celestial information, olfactory signals, and other modalities. This redundancy and integration create robust navigation systems that can function even when individual sensory channels are compromised. But before we explore how these different systems work together, let's look at another fundamental navigation cue that has guided travelers for millennia: the sky itself. Celestial navigation, reading the ancient patterns above. Look up at the night sky from a dark location, away from city lights. You'll see what our ancestors saw: thousands of stars scattered across the darkness, the luminous band of the Milky Way arcing overhead, planets tracing their paths among the constellations. For humans, the stars have been navigation aids for centuries. Sailors learned to find Polaris, the North Star, and calculate their latitude. They learned which stars rose and set at specific points on the horizon, creating a compass rose written in starlight. But humans came to this technique relatively recently. Animals have been reading the sky for millions of years. Let's start with the sun, perhaps the most obvious celestial cue. The sun rises in the east, moves across the sky during the day, and sets in the west. This predictable movement makes it an excellent compass, but there's a complication. The sun moves. Unlike a magnetic field, which points consistently north, the sun's position changes continuously throughout the day. To use it for navigation, an animal needs to compensate for this movement, which requires an internal clock. Many animals have evolved just such a clock, part of their circadian rhythm system. This internal timekeeper, synchronized to the day-night cycle, allows them to know what time of day it is. Combine this time sense with the sun's position, and you can determine direction. Honeybees are masters of sun compass navigation. Carl von Frisch, the pioneering researcher who deciphered the bee waggle dance, demonstrated that bees use the sun as their primary compass. When a forager bee returns to the hive, it performs a dance on the vertical surface of the honeycomb. The direction of the waggle run, relative to vertical, indicates the direction of food relative to the sun's current position. Other bees watching this dance can interpret this information and fly in the correct direction to find the food source. What's elegant about this system is that the dancer bee is performing the dance inside the dark hive, hours after the foraging trip. The dance direction accounts for the sun's movement during the time since the food was found. The bee's internal clock has adjusted the compass bearing automatically. When the watching bees fly out to find the food, they make similar adjustments based on the current sun position. This demonstrates sophisticated time compensation and shows that bees maintain an accurate model of the sun's movement across the sky. But the sun compass has a limitation. It doesn't work on cloudy days or inside dark spaces. Or does it? This is where polarized light comes in. Sunlight becomes partially polarized as it scatters through Earth's atmosphere, creating patterns in the sky that vary predictably with the sun's position. Even when the sun itself is hidden by clouds, these polarization patterns remain detectable. Many insects, including bees, ants, and butterflies, can perceive polarized light through specialized photoreceptors in their eyes. The polarization pattern forms a kind of sky map, with specific patterns associated with different sun positions. By detecting the polarization angle of skylight, animals can determine where the sun is, even when they can't see it directly. This extends the utility of sun compass navigation to overcast days and dawn and dusk when the sun is below the horizon but still illuminating the sky. Desert ants of the genus Cataglyphis make extensive use of polarized light for navigation. These ants forage in the harsh environment of North African and Middle Eastern deserts, where temperatures can soar above 50 degrees Celsius. They navigate across barren salt pans with few visual landmarks. The polarization pattern of the sky provides them with a reliable compass even in these feature-poor environments. Birds use the sun compass extensively during migration. Studies with clock-shifted birds, where researchers kept them in artificial lighting conditions that shifted their internal clocks, have provided compelling evidence. When these clock-shifted birds are released, they fly in directions that are predictably wrong by an amount corresponding to how much their internal clocks were shifted. If a bird's clock is shifted 6 hours late, it will misinterpret the sun's position and fly 90 degrees off course, exactly what you'd expect if it was compensating incorrectly for the sun's movement. But here's the interesting thing. These compass systems need to be learned. Unlike magnetic sensitivity, which appears to have strong innate components, sun compass orientation requires young animals to observe the sun's movement and learn its path across the sky. Young birds that are prevented from seeing the sun in the morning may later have difficulty using the sun for navigation in the afternoon. They haven't learned the complete pattern of the sun's movement. Now let's turn to the stars, those ancient lights that guided mariners long before GPS. For nocturnal migrants, particularly birds that travel at night, the stars provide crucial navigational information. But how does a star compass work when the entire celestial sphere appears to rotate over the course of the night? This is where the research becomes particularly elegant. In the 1960s, Stephen Emlen conducted a series of experiments with indigo buntings, small songbirds that migrate at night. He raised young birds in a planetarium, exposing them to an artificial night sky. Emlen could manipulate this sky, changing which stars appeared and how they moved. What he discovered was remarkable. Birds don't use specific stars or constellations in the way human navigators do. They don't look for Polaris or Orion's belt. Instead, they identify the center of rotation of the night sky. As Earth rotates, the stars appear to wheel around a point in the northern sky near Polaris, the celestial pole. Birds learn to recognize this center of rotation during their first weeks of life, watching the stars move across the night sky. Emlen demonstrated this by showing birds an artificial sky where the stars rotated around a different point, around Betelgeuse instead of Polaris, for example. Birds raised under such conditions would later orient their migration relative to that false center of rotation. The specific pattern of stars didn't matter. What mattered was finding the axis around which the sky rotated. This means the star compass, unlike the sun compass, doesn't require time compensation. The center of rotation stays in the same place regardless of time of night or season. It's a fixed reference point, making it particularly useful for navigation. Birds learn the star pattern during a sensitive period before their first migration. The entire process of calibrating their star compass seems to be driven by watching celestial rotation. Birds that are prevented from seeing a rotating night sky during this critical period later have difficulty using stars for navigation. There's also evidence that some birds can see more than humans when they look at the sky. Many bird species have cone cells in their retinas that are sensitive to ultraviolet light. The night sky may appear richer to them, with more visible stars and perhaps more obvious patterns. Some researchers have speculated that birds might even be able to see auroral emissions or other atmospheric phenomena that help with navigation, though this remains an area of active research. The integration between different celestial cues is also important. At sunset, migratory birds can observe both the sun's setting position and the emerging stars. This gives them an opportunity to calibrate these two compass systems against each other. Studies have shown that birds use the setting sun, along with the polarized light patterns at twilight, to calibrate their magnetic compass. This cross-calibration among different sensory systems, magnetic, solar, and stellar, creates a robust navigation toolkit that maintains accuracy even when individual cues might be compromised. Monarch butterflies provide another stunning example of celestial navigation. These delicate insects migrate thousands of kilometers from Canada and the northern United States to specific mountain valleys in Mexico where they overwinter. Research has shown that monarchs use a sun compass for navigation. Specialized neurons in their brain integrate information about the sun's position with time of day information from their circadian clock, providing directional guidance. Interestingly, no individual monarch completes the round trip. The butterflies that fly south in autumn are several generations removed from the ones that flew north in spring. Yet they find the same wintering sites their great-great-grandparents left months earlier. The navigational program, the ability to use the sun compass and the autumnal migratory direction, is genetically inherited. Dung beetles offer a fascinating twist on celestial navigation. These insects use starlight, even the band of the Milky Way galaxy, for orientation. A dung beetle rolling a ball of dung away from a dung pile needs to move in a straight line to avoid returning to its starting point, where competition is fierce. Studies have shown that on moonless nights, these beetles use the Milky Way as a celestial reference to maintain their bearing, making them the only known insects to orient using the pattern of our galaxy. The use of polarized moonlight by some beetle species adds another layer to celestial navigation. The moon, like the sun, creates polarization patterns in the sky, though much weaker. Nonetheless, some nocturnal insects have evolved sufficient sensitivity to use these patterns for orientation. What's important to understand is that celestial navigation, like magnetic navigation, is rarely used in isolation. Diurnal migrants may start their flights using sun compass orientation, then switch to magnetic compass information if clouds obscure the sun. Nocturnal migrants may use stars to set their initial heading at dusk, then rely on magnetic cues during the night, potentially recalibrating against stars if cloud cover clears. This flexibility, this ability to switch between navigation systems depending on availability and reliability, is a hallmark of sophisticated animal navigators. But not all navigation cues come from grand cosmic sources like Earth's magnetic field or the movement of celestial bodies. Some of the most powerful navigation signals are far more subtle, carried on the wind as invisible molecules, detected by senses we often underestimate. Let's explore the world of chemical navigation. Chemical wayfinding, following invisible trails. Imagine standing beside a river, watching salmon leap up a waterfall, hurling themselves against the rushing water, again and again until they succeed or exhaust themselves trying. These fish have traveled from the ocean, sometimes swimming thousands of kilometers, and are now fighting their way upstream to the exact stretch of river, sometimes the precise gravel bed where they hatched years earlier. How do they know where to go? The answer is scent, smell, chemical molecules dissolved in water, detected by sensory systems so refined they can identify the unique chemical signature of a specific stream among thousands. Chemical navigation, or olfactory navigation, is ancient and widespread in the animal kingdom. For aquatic animals, water is an excellent medium for carrying chemical information. Molecules dissolve, diffuse, and create trails and plumes that can be followed upstream to their source. For terrestrial animals, air carries volatile chemicals that can travel long distances on wind currents. An animal with the right receptors can detect these molecules and use them for navigation. Salmon are perhaps the most famous example of olfactory navigation. Pacific salmon species, including Chinook, coho, sockeye, pink, and chum, are all anadromous, meaning they hatch in freshwater, migrate to the ocean to feed and grow, then return to freshwater to spawn. The accuracy of their homing is remarkable. Studies have shown that salmon can return not just to the same river system, but often to the same tributary, even the same section of stream where they began life. This ability is based on olfactory imprinting, a process where juvenile salmon learn and remember the chemical signature of their natal water. During a critical period in their development, particularly during the pare-smolt transformation, when they're preparing to migrate to sea, young salmon imprint on the specific mixture of chemicals in their home stream. These chemicals come from various sources: the surrounding geology and soil, the vegetation, other organisms in the water, and even the metabolic waste products and pheromones from their own kin. The imprinting process appears to involve changes in gene expression in the olfactory epithelium, the sensory tissue in the salmon's nose. Research has shown that specific olfactory receptor genes become more active during the smolt stage. Hormones, particularly thyroid hormones and salmon gonadotropin-releasing hormone, play crucial roles in preparing the olfactory system for imprinting and later in reactivating these memories when adult salmon return to spawn. The evidence for olfactory imprinting comes from several types of experiments. In one classic study, researchers exposed juvenile coho salmon to a synthetic chemical called morpholine, which doesn't occur naturally in their environment. Years later, when these fish returned as adults to spawn, morpholine was released into one branch of the river system. The exposed fish overwhelmingly chose the morpholine-scented stream, even though they had never experienced that chemical in their natal water. The fish had been successfully imprinted on an artificial odor and used it for navigation. Field studies tracking wild salmon have provided complementary evidence. When scientists have blocked or manipulated the salmon's sense of smell by plugging their nares, the olfactory organs, the fish lose much of their ability to navigate accurately to their home stream. They still show some homing ability, likely using other cues, but their precision is significantly reduced. The chemical composition of a stream is like a fingerprint. It results from a unique combination of factors: the minerals in the bedrock, the plants growing along the banks, the insects and other invertebrates living in the water, the surrounding soil composition, even the pattern of rainfall and snowmelt. Each stream has its own chemical signature, stable enough to be recognizable year after year, yet distinct enough to be differentiated from other streams. But here's a challenge. The chemical signature of a stream isn't static. It changes with seasons, with weather patterns, with floods that scour the streambed, or droughts that concentrate dissolved minerals. How can salmon use a chemical memory imprinted years ago when they were juveniles to navigate to a stream whose chemistry may have changed? The answer appears to be that salmon don't imprint on every individual chemical compound. Instead, they learn a pattern, a gestalt of multiple chemicals that together define their home stream. Even if some components change, the overall pattern remains recognizable. It's like how you can recognize a person's voice even when they have a cold, or recognize a piece of music even if it's played on a different instrument. The fundamental pattern persists despite surface variations. Recent research has also suggested that salmon may use a hierarchical strategy for homing. At sea, far from any river, they likely use magnetic information and ocean currents to navigate to the general coastal area where their natal river empties. As they approach the coast and enter water influenced by river outflow, they begin to use olfactory cues, testing the water for familiar chemical signals. The closer they get to home, the more they rely on olfactory information, eventually using it as their primary guide through the river system to their spawning grounds. The neural mechanisms underlying this behavior are beginning to be understood. The olfactory system of salmon is remarkably sophisticated. They possess multiple types of olfactory receptors, each tuned to detect different classes of chemicals. When a salmon encounters a familiar odor, specific combinations of these receptors are activated, creating a pattern of neural activity that the brain can recognize. This recognition appears to happen in the olfactory bulb, where sensory information from the nose is first processed, and in higher brain areas, including the telencephalon, the fish equivalent of the mammalian cerebral cortex. Studies measuring electrical activity in these brain regions have shown that salmon demonstrate heightened responses to water from their home stream compared to water from other locations, even when the chemical differences are subtle. But olfactory navigation isn't limited to salmon. Many other fish species use chemical cues for homing. Studies with sharks and rays have shown that they can detect and orient to waterborne odors from considerable distances. Eels, which make extraordinary migrations from freshwater rivers to spawning areas in the Sargasso Sea, are thought to use olfactory information as part of their navigation strategy, though the details remain somewhat mysterious. Moving to land, olfactory navigation becomes more challenging, but is still used by many species. The chemical signals are more ephemeral, carried by wind, dispersed by turbulence, degraded by sunlight and oxidation. Yet, terrestrial animals have evolved to exploit these cues. Homing pigeons, surprisingly, may use olfactory information as part of their navigation toolkit. Research in Italy has shown that pigeons prevented from smelling by having their olfactory nerves cut or their nares sealed show impaired homing ability, particularly when released from unfamiliar locations. The hypothesis is that pigeons learn an olfactory map of their home area, associating different smells with different wind directions. By noting which smells are strongest in their home loft, they can later determine their position relative to home by sampling the air at a release site and noting which familiar smells are present. This idea remains somewhat controversial, and the mechanism isn't fully understood, but it highlights an important point. Navigation systems can be multimodal, using whatever sensory information is available and reliable. A pigeon might use the sun compass as its primary navigation tool, but supplement this with olfactory information, magnetic cues, and visual landmarks. Sea turtles also appear to use olfactory cues as part of their navigation strategy. When approaching coastal areas during their breeding migrations, female sea turtles likely use the scent of their natal beach, carried in the water, to help locate the specific nesting site. The chemical signature might come from the surrounding vegetation, the particular sand composition, or even pheromones from other turtles. While magnetic cues can guide them to the general area, olfactory information may provide the fine-scale guidance needed to identify the exact beach. Insects provide some of the clearest examples of olfactory trail following. Ants lay down pheromone trails between their nest and food sources. An individual ant discovering food returns to the nest while depositing a chemical trail. Other ants can follow this trail to the food, reinforcing it with their own pheromone deposits. The result is a positive feedback system where heavily used trails become stronger, creating efficient networks of chemical highways between the colony and valuable resources. Male moths use olfactory cues to locate females, detecting pheromones at extremely low concentrations. A silk moth can detect and orient to a single molecule of bombicol, the female's sex pheromone, in a room-sized space. The moth flies upwind when it detects the pheromone, using the wind direction to guide it toward the source. What's particularly sophisticated about moth olfactory navigation is the strategy called casting. When the moth loses the pheromone trail, it begins flying in a zigzag pattern, increasing its chances of re-encountering the chemical plume. Once it detects the pheromone again, it resumes flying upwind. This behavior demonstrates active problem-solving in response to interrupted sensory information. The challenge with olfactory navigation is that chemical plumes and trails are ephemeral and variable. Wind patterns change, currents shift, chemicals degrade. An olfactory navigator must be able to deal with this variability, interpreting partial information and making decisions based on probabilistic, rather than certain, data. This is where the integration of multiple sensory modalities becomes crucial. An animal using olfactory cues might combine this information with visual landmarks, path integration, or magnetic cues. The olfactory signal might confirm location or direction suggested by other senses, or it might provide the deciding information when other cues are ambiguous. The study of olfactory navigation has practical implications for conservation. For salmon, maintaining the chemical integrity of natal streams is crucial for successful homing. Pollution, changes in land use, alteration of streamside vegetation, all of these can modify the chemical signature of a stream and potentially impair the ability of salmon to find their way home. Hatchery programs that raise salmon in unnatural conditions may not allow proper imprinting, contributing to lower homing success and genetic mixing between populations. For sea turtles, light pollution on nesting beaches can overwhelm natural olfactory cues, disorienting females and preventing successful nesting. Understanding how these animals use chemical information for navigation informs conservation strategies designed to protect not just the animals themselves, but the sensory environment they depend on. As we move forward in our exploration, keep in mind that olfactory navigation, like the other systems we've discussed, is rarely used in isolation. It's one tool in a varied toolkit, valuable for certain situations and supplemented by other senses when necessary. Now let's turn to a completely different navigation strategy, one that uses sound instead of sight, smell, or magnetism. Let's explore the world of echolocation. Sound as a guide, echolocation in the darkness. Imagine flying through a pitch-black cave at high speed, maneuvering around rock formations, avoiding other fast-moving creatures, and precisely snatching flying insects out of the air, all without seeing anything. Or imagine swimming through murky coastal waters, where visibility is measured in centimeters, detecting a fish the size of your thumb from 100 meters away, determining not just its location, but its size, speed, and species. This is the world of echolocation, where animals navigate and hunt using sound. Echolocation is biological sonar. An animal emits a sound, usually at frequencies too high for humans to hear, and listens for the echoes that bounce back from objects in the environment. By analyzing these echoes, particularly the time delay between emission and return, the animal can determine the distance to the object. By analyzing how the echo's frequency or intensity changes, the animal can determine the object's movement, size, texture, and material composition. Two groups of mammals have independently evolved sophisticated echolocation: bats and toothed whales, including dolphins. The fact that these systems evolved independently in animals as different as a 3-gram bat and a 3-ton whale speaks to the power and utility of echolocation as a navigation and hunting strategy. Let's start with bats, those mysterious creatures of the night. Most bat species echolocate, though a few, particularly large fruit bats, rely primarily on vision. Echolocating bats produce ultrasonic calls, sounds with frequencies above the human hearing range of about 20,000 Hz. Some bat calls reach frequencies of 200,000 Hz, nearly 10 times higher than what humans can hear. The calls are produced in the larynx, much like vocal sounds in humans, but at frequencies and intensities we could never achieve. The sound is then emitted either through the mouth or the nose, depending on the species. Bats with elaborate nose structures, called nose leaves, emit their echolocation calls through their nostrils, with the nose leaf acting to focus and direct the sound beam. The calls themselves are remarkably varied. Some bats produce brief clicks, lasting just a few milliseconds. Others produce longer calls with constant frequency, sometimes called CF calls, that can last tens of milliseconds. Many bats use frequency-modulated calls, or FM calls, where the pitch sweeps rapidly from high to low. Some species use combinations of CF and FM components in a single call. The type of call a bat uses depends on its hunting strategy and environment. In cluttered environments like dense forests, short FM calls work best. The rapid frequency sweep provides excellent information about an object's distance and is less likely to produce confusing echoes from multiple surfaces. In open environments, longer calls that travel farther are advantageous, allowing the bat to detect prey at greater distances. The precision of bat echolocation is extraordinary. A bat can detect a wire as thin as a human hair in complete darkness. When closing in on flying insect prey, some bats can detect movement on the scale of the insect's wing beats and predict where the insect will be, leading their target like a fighter pilot. But here's one of the challenges of echolocation. The bat must distinguish the faint echo of its call, returning from a tiny, distant object from the much louder sound of its own call. This is like trying to hear a whisper while someone's shouting in your ear. Bats solve this problem with a remarkable adaptation. They temporarily shut down their hearing while vocalizing. Tiny muscles in the middle ear contract just before each call, dampening the sound reaching the inner ear. Then, within a few milliseconds, the muscles relax and hearing is restored, just in time to hear the returning echo. This happens dozens or even hundreds of times per second when a bat is hunting. As the bat approaches its target, it increases its call rate in what's known as a terminal buzz, producing calls so rapidly they blend into a buzz sound if played in a frequency range humans can hear. This acceleration allows the bat to update its information about the prey's location more frequently as the critical moment of capture approaches. The neural processing underlying bat echolocation is sophisticated. The bat's brain has regions specifically dedicated to analyzing echolocation signals. Neurons in the auditory cortex are tuned to specific combinations of call frequency, echo delay, and Doppler shift. The change in frequency caused by relative motion between the bat and its target. This creates a neural map of the environment based on acoustic information. Some bat species, particularly horseshoe bats and old-world leaf-nosed bats, have evolved specialized echolocation for detecting flying insects against background clutter. They emit long CF calls at a very specific frequency, usually matched to a frequency where their hearing is most acute. When an insect's wings beat, they create periodic changes in the echo's frequency through the Doppler effect. The bat can detect these flutter patterns and distinguish a flying insect from stationary vegetation, even when the insect is very close to leaves or other surfaces that would create confusing echoes. Now, let's turn to echolocation in the ocean, where sound travels nearly 5 times faster than in air and much farther. Toothed whales and dolphins have evolved echolocation systems that are, in some ways, even more sophisticated than those of bats. Dolphins produce echolocation clicks using specialized structures in their nasal passages, not their larynx. The clicks are produced in a complex of air sacs and passages near the blowhole. These clicks are then projected forward through the melon, a large rounded structure filled with specialized fats in the dolphin's forehead. The melon acts as an acoustic lens, focusing the sound into a directional beam that the dolphin can aim by moving its head. A bottlenose dolphin can detect a fish the size of a ping-pong ball from over 100 meters away, far beyond what it could see in the often turbid coastal waters where many dolphins live. The clicks are extremely short, lasting only microseconds, and at very high frequencies, up to 150,000 Hz. This gives the echoes excellent temporal resolution, meaning the dolphin can detect fine details about the object's structure. When a dolphin echolocates on a person, it's been described as feeling like a physical sensation, a buzzing or tingling. The dolphin is essentially scanning you with sound waves, seeing through your skin to perceive your skeleton, organs, perhaps even your circulatory system. To a dolphin, you're somewhat transparent, like you are to an ultrasound machine. The returning echo is received not through the dolphin's external ears, which are tiny holes behind the eyes, but through the lower jaw. The lower jaw contains specialized fats that conduct sound to the inner ear. This allows the dolphin to determine not just that an echo has returned, but also the direction from which it came. Essential information for localizing an object in 3-dimensional space. Like bats, dolphins adjust their echolocation behavior based on the situation. When searching for prey in open water, they produce clicks at a relatively slow rate. As they approach a target, the click rate increases, sometimes reaching hundreds of clicks per second. They also increase the intensity of their clicks in noisier environments, or when trying to detect distant targets. Research has shown that dolphins can determine an extraordinary amount of information from echoes. They can distinguish between objects of different materials, identifying a copper disc versus an aluminum disc of the same size. They can determine the internal structure of objects, recognizing a hollow ball versus a solid ball. They can even detect the size and thickness of fish from the acoustic properties of the echo. Sperm whales have the largest brain of any animal, and a significant portion of it is dedicated to acoustic processing. These deep-diving whales hunt squid in complete darkness, sometimes at depths exceeding 2,000 meters. They produce extremely loud echolocation clicks, among the loudest sounds created by any animal. These clicks are produced in the spermaceti organ, a huge structure filled with wax-like oil that occupies much of the whale's massive, squared-off head. The clicks produced by sperm whales are so loud they could theoretically damage tissue. Researchers working with captive dolphins have measured click intensities that would be dangerous if directed at a human at close range. In the wild, there have been unconfirmed reports of divers feeling stunned or disoriented when echolocating dolphins or small whales have swum too close. Whether these animals can use their echolocation as a weapon, stunning prey with intense sound, remains a topic of debate, but it's clear that the acoustic energy involved is substantial. One fascinating aspect of cetacean echolocation is that the animals can do it continuously while swimming, navigating, and hunting. They maintain an ongoing acoustic picture of their surroundings. Unlike humans using sonar on a submarine, who must interpret blips on a screen, a dolphin directly perceives the acoustic environment as an integrated sensory experience, probably not too different from how we experience vision. The convergent evolution of echolocation in bats and dolphins is remarkable, not just in the gross similarity of the systems, but in the molecular details. Researchers have found that the same genes underwent similar mutations in both bats and dolphins, mutations that affect hearing, and particularly the ability to process high-frequency sounds. This is one of the clearest examples of convergent evolution at the genetic level, where natural selection has produced similar solutions to similar problems independently in very different animals. A few other animals have evolved forms of echolocation, though generally less sophisticated than in bats and dolphins. Some species of cave-dwelling swiftlets and the oilbird use echolocation to navigate in dark caves where they roost. They produce audible clicks in the range humans can hear, and use the echoes to avoid colliding with cave walls. Some shrews and tenrecs, small insect-eating mammals, produce ultrasonic clicks that may serve an echolocation function, though this is less well studied than in bats. Even some humans have developed echolocation abilities. Blind individuals who learn to make clicking sounds with their tongue, or by snapping their fingers, can navigate environments with remarkable skill, detecting walls, doorways, and objects by listening to the echoes. Brain imaging studies have shown that the visual cortex of echolocating blind people activates in response to echoes, suggesting that the brain can repurpose visual processing areas for acoustic spatial information. The evolution of echolocation has allowed animals to exploit niches that would otherwise be inaccessible. Bats have become the dominant nocturnal aerial insect hunters, avoiding competition with birds and escaping many predators that rely on vision. Dolphins and other toothed whales can hunt effectively in conditions where vision is useless, from murky coastal waters to the eternal darkness of the deep sea. But like other navigation systems, echolocation isn't used in isolation. Bats combine echolocation with vision when there's enough light. Dolphins use echolocation along with vision, hearing passive sounds, and potentially magnetic sensing for long-distance navigation. The acoustic information complements and enhances what other senses provide. Now, let's shift from the sophisticated technology of biological sonar to something that seems, on the surface, much simpler, but turns out to be remarkably complex. Let's explore how animals keep track of their movements through space using only internal information, a process called path integration. The ancient art of path integration, navigation by dead reckoning picture. A desert ant emerging from her nest in the morning. The nest entrance is a small hole in the ground, perhaps a centimetre across, in an expanse of barren sand dunes that stretches to every horizon. There are no landmarks to speak of. The temperature is already climbing toward 40 degrees Celsius. The ant sets out to search for food. She wanders in a tortuous, zigzagging path, searching for dead insects, wandering sometimes 100 meters from the nest, taking a route that twists and turns, doubling back on itself. After an hour of searching, she finds the corpse of a beetle. She grabs it and, without hesitation, turns and walks in an almost perfectly straight line back toward her nest. Not toward where the beetle was found, not retracing her outbound path, but in a direct line, the shortest route, straight back home. How? She can't see the nest. She can't smell it from this distance. She's never traveled this exact route before. Yet she knows, with remarkable precision, both the direction and the distance to her nest. This is path integration, also called dead reckoning, one of the most fundamental navigation mechanisms in the animal kingdom. The principle is elegant. Keep track of every movement you make, direction and distance, and continuously update a running calculation of your position relative to your starting point. The result is a home vector, an internal representation of the straight line route back home. Path integration requires two pieces of information: directional information, which way have I been moving, and distance information, how far have I gone. Different animals obtain these using different mechanisms, but the computational principle is the same. For desert ants, Cataglyphis fortis is one of the most studied species. Direction comes primarily from the sun. But remember, the sun moves across the sky, so ants, like bees, use the sun along with their internal clock to determine compass direction. They compensate for the sun's movement, maintaining an accurate heading even during a foraging trip that lasts hours. When the sun isn't visible, ants use polarized light patterns in the sky. Even when clouds obscure the direct sunlight, the polarization pattern often remains detectable, providing directional information. Research has shown that Cataglyphis ants have specialized photoreceptors in a region of their compound eyes called the dorsal rim area, specifically adapted for detecting polarization angles. But direction alone isn't enough. The ant also needs to know how far she's traveled in each direction. This is where things get really interesting. For many years, scientists debated how ants measured distance. Did they count steps? Did they measure time? Did they use visual cues, measuring how much the image of the ground moved past their eyes, what's called optic flow? A brilliant experiment settled the question. Researchers captured ants as they returned from foraging trips and modified their legs. In one group, they glued tiny stilts onto the ants' legs, making them taller. In another group, they cut the ants' legs shorter. Then, they released the ants and watched where they searched for the nest entrance. The ants on stilts walked past the nest entrance, searching for it farther away than it actually was. The ants with shortened legs stopped short, searching before they'd reached the nest. The stilted ants took longer strides and thus took fewer steps to cover the distance. Their step counter told them they'd walked less far than they actually had. The opposite was true for the ants with shortened legs. This experiment demonstrated that ants use a stride integrator, essentially a step counter, to measure distance. With each step, a small amount is added to their internal distance measure. The neural mechanism likely involves proprioceptors, sensors that detect the position and movement of the legs, though the exact details are still being investigated. What's remarkable is the precision. Desert ants can forage over distances of hundreds of meters and return to a nest entrance just a few millimeters wide, all based on their path integration system. The accuracy degrades with distance, as small errors in direction or distance measurement accumulate. But it's still impressively precise. Path integration becomes even more impressive when you consider that desert ants can navigate in 3 dimensions. When an ant climbs over a rock or hill, it doesn't just measure the distance traveled along the sloped surface. It calculates the horizontal distance, the projection onto the ground plane. So, if an ant walks up a 30-degree slope for 10 meters, it registers this as approximately 8.7 meters of horizontal distance, not 10 meters. How does a creature with a brain smaller than a grain of sand perform this geometric calculation? Researchers are still working out the details, but it appears the ants can measure the inclination of the path and incorporate this into their distance calculation. This is computational feat that would require trigonometry if we did it explicitly, yet the ant accomplishes it automatically. But path integration isn't limited to insects. Birds use it during migration, when flying over featureless ocean, or in overcast conditions where celestial cues aren't available. Birds can maintain their heading and track their displacement using path integration. The compass information might come from magnetic cues, and the distance information might come from motor efferents. The brain's awareness of the movement commands its issuing, or from vestibular information, the sense of acceleration and movement from the inner ear. Mammals, including humans, use path integration. When you walk through a dark room, you maintain a sense of where you are relative to where you started. This is path integration. It's less precise in humans than in desert ants, but the principle is the same. Studies with rodents have provided insights into the neural mechanisms underlying path integration. The hippocampus, a brain structure crucial for spatial memory, contains cells called grid cells and place cells that appear to encode spatial position and that update their activity based on the animal's movement. Grid cells, discovered in rats, fire when the animal is at specific locations in the environment. But these locations are arranged in a regular, repeating triangular grid. As the rat moves, the active grid cells update based on the rat's movement, effectively performing path integration at the neural level. Place cells provide a complementary representation, with each cell firing at a specific place in the environment. Together, these cell types may form the basis of the mammalian path integration system. Path integration has some important limitations. Because it relies on integrating many small measurements, errors accumulate. A small directional error, repeated over many steps, leads to increasingly large positional errors. This is why animals that rely heavily on path integration also use other navigation strategies to correct these accumulated errors. For desert ants, visual landmarks, when available, can reset the path integrator. When an ant recognizes a familiar place through visual cues, it can correct its estimate of its position. This combination of path integration, providing continuous positional updates, and landmarks, providing occasional corrections, creates a robust navigation system. Research has also shown that ants can store multiple path vectors simultaneously. An ant that has learned the locations of 2 feeding sites can travel between them directly, calculating the necessary vector by combining the stored home vectors for each site. This suggests a more flexible use of path integration than simple dead reckoning. The computational principles of path integration are so fundamental that they've been incorporated into some robotic navigation systems. Engineers building autonomous robots have looked to desert ants and other path-integrating animals for inspiration. implementing similar strategies for allowing machines to track their position when GPS isn't available. One fascinating aspect of path integration is its reliability under challenging conditions. Desert ants navigate successfully even in extreme heat, even when their body temperature is high enough that it would be fatal if sustained for longer. The path integration system continues to function accurately, suggesting it's implemented in neural circuits that are robust to thermal stress. The study of path integration has also revealed interesting things about how animals prioritize different navigation cues. When researchers have put path integration in conflict with other navigation systems by displacing ants during their return journey, for example, the ants initially try to run off their home vector. They do what path integration tells them, even though this doesn't lead them home. Eventually, when they don't find the expected landmarks, they switch to systematic searching. This reveals a hierarchical organization of navigation systems. Path integration provides a baseline, an always available default strategy. Other cues, like landmarks or magnetic information, can override or correct this baseline when they're available and when the animal has learned to use them. But path integration, for all its elegance, is just one tool in the navigation toolkit. Let's now turn to a very different strategy, one that depends not on internal calculation, but on external reference points. The use of visual landmarks and the formation of cognitive maps, visual landmarks and memory maps, navigating a remembered world. Close your eyes and think about your route home. Can you picture it? Perhaps you see specific buildings, a distinctive tree, the corner where you turn. These are landmarks, memorable features of the environment that you use to determine where you are and where you need to go. Visual landmarks are one of the oldest and most intuitive navigation strategies, used by animals ranging from insects to humans. But landmarks can be used in different ways, and the sophistication with which animals use them varies considerably. At the simplest level, a landmark can act as a beacon, a cue that marks the goal location directly. A bee might remember that food is located at a yellow flower. Flying to yellow flowers becomes the navigation strategy. This is straightforward, effective for short-range navigation, but limited. More sophisticated landmark use involves remembering the spatial relationships between multiple landmarks and using these relationships to determine position and navigate to goals that may not be directly visible. This is where we start to see evidence of cognitive maps, internal representations of space that allow flexible navigation. Let's start with a fascinating example, the waggle dance of honeybees, which we touched on earlier, but deserves deeper exploration. When a forager bee returns from a successful trip and performs a waggle dance, she's encoding the location of food relative to the hive and the sun. The dance direction indicates the angle between the sun and the food source. The duration of the waggle run indicates the distance. But here's what makes this remarkable. The bee is dancing inside the dark hive, on a vertical surface, hours after finding the food. She's translating a remembered location in the horizontal plane, outside the hive, into a dance on a vertical surface, adjusting for the sun's movement since she last saw the food, and doing so in a way that other bees can interpret and use to find the food themselves. This is sophisticated spatial cognition. The bees watching the dance gain information about a location they've never visited. They then fly out and navigate to that location, using a combination of their sun compass to maintain the correct direction and their own path integration to gauge the distance. If they're familiar with the area, they might also use remembered visual landmarks to guide their approach. Research has shown that bees can learn and remember multiple landmark configurations. In experiments where bees are trained to find food in relation to an array of visual cues, they demonstrate that they've learned the entire spatial arrangement, not just individual landmark identities. If the landmarks are moved as a group, the bees search in the correct location relative to the moved landmarks. If individual landmarks are moved independently, disrupting the spatial relationships, the bees show confusion. This suggests that bees store snapshot memories of what the world looks like from specific locations, particularly from goal locations like the nest entrance or a productive flower patch. When returning to these locations, bees attempt to match their current view to these stored snapshots, moving in directions that increase the match. This view-based navigation is computationally efficient and appears to be used by many insects. Desert ants also use visual landmarks, though in their harsh environment, landmarks may be sparse. Studies have shown that cataglyphis ants can learn the appearance of individual rocks or plants and use these as navigation cues. Importantly, ants learn landmarks in relation to their path integration system. As they walk away from the nest, they store views of the environment at specific distances and directions from home, as indicated by their path integrator. When returning, they can recognize these familiar views and use them to confirm or correct their path integration estimate. Birds provide examples of even more sophisticated landmark use. Homing pigeons, which have been studied extensively, learn visual maps of their home area during exploratory flights early in life. When released from a distant, unfamiliar location, they use other cues, like the sun compass or magnetic information, to determine the general direction home. As they approach familiar territory, they transition to using memorized visual landmarks for more precise navigation. Interestingly, research has shown that pigeons learn different types of information at different spatial scales. For long-distance homing, they may rely on large-scale cues, like the position of mountain ranges or coastlines. For finding their specific loft within a familiar city, they rely on more local landmarks, like buildings and streets. This hierarchical representation, where different levels of spatial detail are encoded separately, is efficient and flexible. Brain imaging studies and lesion experiments have provided insights into the neural basis of landmark-based navigation. In mammals, the hippocampus is crucial for spatial memory and landmark learning. Rats with hippocampal damage can still perform path integration and can navigate to visible beacons, but they're impaired at tasks requiring memory of spatial relationships between landmarks. The hippocampus contains place cells, neurons that fire when the animal is in specific locations in an environment. These place cells seem to encode a map-like representation of space, with different cells active at different locations. Importantly, place cell activity is strongly influenced by visual landmarks. When landmarks are rotated, place cell representations rotate correspondingly, showing that landmarks control the spatial reference frame. More recently, scientists have identified grid cells in the entorhinal cortex, a region that provides input to the hippocampus. Grid cells fire at multiple locations in an environment, but these locations are arranged in a regular, repeating hexagonal pattern. The spacing of this grid varies systematically, with some grid cells having fine spacing and others coarse spacing. This creates a multi-scale representation of space that may allow precise position encoding over large areas. The interaction between place cells, grid cells, and landmark information is thought to underlie the cognitive mapping abilities of mammals. Path integration, mediated by grid cells, provides a continuous estimate of position. Landmark recognition, mediated by visual processing areas and place cells, provides corrections to this estimate. The result is a robust spatial representation that combines the advantages of path integration, always available, continuous updates with the advantages of landmark use, high precision, error correction. Some researchers have debated whether insects possess cognitive maps in the same sense that mammals do. The question hinges on what we mean by a map. If a map is an internal representation that allows flexible navigation, including shortcuts between known locations, then there's evidence that at least some insects have map-like representations. Experiments with ants have shown that individuals trained to visit 2 feeding sites can sometimes take shortcuts between these sites, suggesting they've formed some kind of integrated spatial representation. However, other experiments have shown that much of what appears to be map-like behavior in insects can be explained by the interaction of path integration and stored views, without requiring a map in the sense of a unified, metric representation of space. This debate highlights an important point. There may be multiple ways to achieve flexible navigation. Mammals with large brains and complex hippocampal structures may use one solution. Insects with tiny brains may use a different, more distributed solution that achieves similar results through the interaction of simpler mechanisms. Visual landmark navigation, like other navigation strategies, has limitations. It works best in environments rich with visual features. In featureless environments like the open ocean, the deep sea, or flat deserts, visual landmarks are scarce or absent. In dense forests, visual landmarks may be hidden by vegetation. In these situations, animals must rely more heavily on other navigation cues. There's also the challenge of view dependence. If an animal learns the appearance of landmarks from one direction, will it recognize them from other directions? Some animals show view-dependent memory, where they're better at recognizing landmarks from familiar viewpoints. Others show more view-invariant recognition, suggesting they extract and remember more abstract features of landmarks. The integration of landmark information with other navigation cues is crucial for robust navigation. An animal might use path integration to maintain an estimate of its position, use landmarks to correct this estimate when they're available, and use magnetic or celestial cues to orient when landmarks are out of view. This kind of sensor fusion, combining information from multiple sources, is a hallmark of sophisticated navigation systems. As we've explored these various navigation strategies, path integration, landmarks, celestial cues, magnetic sensing, olfaction, echolocation, you might have noticed a recurring theme. These systems don't work in isolation. Animals combine multiple sources of information, using each where it's most reliable and integrating them into a coherent understanding of where they are and where they're going. Now let's look at a specific example that beautifully illustrates this multimodal approach, the remarkable navigation of sea turtles. Sea turtles, masters of multimodal navigation. It's a moonlit night on a beach in Florida. In the sand, barely visible in the darkness, movement. Tiny flippers push upward. One by one, loggerhead sea turtle hatchlings emerge from their nest, where they've been incubating for the past 60 days. Each is about the size of a cookie, weighing less than an ounce. They've never seen the ocean. They've never seen anything. Yet, they immediately orient toward the water and begin crawling. What happens next is one of the most extraordinary journeys in the animal kingdom. These hatchlings will swim away from shore and enter a vast ocean gyre, the North Atlantic subtropical gyre, which will carry them on a multi-year journey covering thousands of miles. They'll circle the Atlantic Ocean, passing by the Azores, down the coast of Africa, across to South America, and back toward Florida. And if they survive, which most won't, the females among them will return to nest on a beach very close to where they hatched, completing the cycle. How do these tiny turtles, with brains the size of a pinhead, navigate across an ocean? The answer involves nearly every navigation mechanism we've discussed, layered and integrated in sophisticated ways. Let's follow a hatchling's journey and see which navigation cues come into play at each stage. The first challenge is simply finding the ocean. The hatchlings emerge from the nest at night, an instinctive behavior that helps them avoid predators and the heat of the day. But it's dark. How do they know which way to go? The answer is light. The ocean, on a natural beach, is brighter than the land. The open sky over the water is less obstructed than the vegetated dunes behind the beach. Moonlight and starlight reflecting off the water create a bright horizon. The hatchlings orient toward this brightness, scrambling down the slope of the beach toward the water. This is where human activity creates problems. Artificial lights from beachfront development can be brighter than the natural ocean horizon, causing hatchlings to crawl inland toward roads and buildings instead of toward the sea. This is a perfect example of how human modifications to the environment can disrupt natural navigation cues. But light isn't the only cue for sea finding. Research has shown that hatchlings also orient downslope, toward lower elevation. Even if disoriented by artificial lights, this downslope orientation can sometimes guide them to the water. They're combining multiple cues, visual and gravitational, to solve the sea-finding problem. Once in the water, the hatchlings face a new challenge: swimming away from shore. Staying in shallow coastal waters is dangerous. Predators are abundant. Currents can push them back onto the beach, and they need to reach the open ocean to begin their migratory circuit. Here, a different cue comes into play: wave direction. As waves approach a beach, they refract, bending until they're moving nearly perpendicular to the shoreline. Hatchlings can detect the direction of these approaching waves. They dive beneath the surface and sense the orbital water motion created by the waves. By swimming directly into the approaching waves, they reliably swim away from shore. Experiments have demonstrated this beautifully. Researchers placed hatchlings in a tank where they could manipulate wave direction using wave machines. The hatchlings consistently oriented into the artificial waves, confirming that they use this cue. In the wild, this strategy works reliably because wave refraction ensures that swimming into waves will take you offshore. But waves are only useful in shallow water. Once the hatchlings reach deeper water, perhaps a few kilometers from shore, wave orientation becomes less reliable. Now they transition to a different navigation system, the magnetic compass. This is where it gets really interesting. Hatchling loggerheads have an inherited magnetic compass. They know, without any previous experience, which magnetic direction is safe. Florida loggerheads, when placed in magnetic fields, characteristic of different parts of the Atlantic, orient in directions that would keep them within the North Atlantic gyre. If exposed to magnetic fields from the northern part of the gyre, they swim south. If exposed to fields from the southern part, they swim north or northeast. They're responding to magnetic information they've never experienced before, following a genetically programmed navigational map. This inherited magnetic compass is tuned to the specific oceanic region where the turtles develop. It keeps them within favorable waters, preventing them from straying into regions that are too cold or too far from eventual return routes. It's pre-programmed knowledge, installed by millions of years of natural selection on populations that stayed within the gyre versus those that wandered into unsuitable areas. But the sophistication doesn't stop there. As the young turtles grow and develop, spending years in the open ocean, they transition from using this simple inherited compass to developing a learned magnetic map. They begin to recognize specific magnetic signatures, combinations of field intensity and inclination that mark particular regions of the ocean. This learned map allows more flexible and precise navigation. By the time they're juveniles, sea turtles can determine their approximate position in the ocean by sensing the local magnetic field. Research has shown that turtles can detect differences in magnetic inclination of less than a degree and differences in intensity too small for human-built compasses to easily measure. This gives them what's essentially a bi-coordinate magnetic map, where each location has a unique magnetic address. Adult sea turtles use this magnetic map for remarkable feats of navigation. Green sea turtles, foraging around the Galapagos Islands, travel to nesting beaches on Ascension Island, a tiny island in the middle of the Atlantic Ocean, over 2,000 kilometers away. They find it with high precision, suggesting they're using their magnetic map to determine both their starting position and Ascension's position, then calculating and following the appropriate heading. But even with a sophisticated magnetic map, there's a final stage where other cues come into play. As an adult female approaches her natal beach to nest, magnetic information can get her to the right stretch of coastline. But how does she find the specific beach where she hatched decades earlier? This is where olfactory cues likely become important. Research suggests that turtles can detect chemical signatures in the water, perhaps from specific vegetation, soil, or even pheromones from other turtles. These chemical cues provide fine-scale guidance, allowing the turtle to identify the exact beach within a larger coastal area that has similar magnetic properties. There may also be a role for visual landmarks, though this is less certain. Turtles have good vision, and they might recognize coastal features, the shape of a bay, the profile of hills behind the beach, that help them identify their natal area. The combination of magnetic information, olfactory cues, and possibly visual landmarks creates a robust system for natal homing. The really remarkable thing about sea turtle navigation is how it develops over the animal's lifetime. The hatchling uses simple cues: light, slope, and wave direction to reach the ocean, then an inherited magnetic compass to stay in safe waters. The juvenile develops a learned magnetic map, transforming from following innate responses to using learned spatial knowledge. The adult integrates this magnetic map with chemical and possibly visual cues for precise homing. It's an ontogenetic progression, from simple to sophisticated, from innate to learned, from single cue to multimodal navigation. This multimodal approach provides redundancy and robustness. If one cue is temporarily unavailable, perhaps clouds obscure visual landmarks, or changing currents alter chemical plumes, the turtle can rely on other cues. The system degrades gracefully, rather than failing completely. Research on sea turtle navigation has important conservation implications. Understanding that turtles imprint on the magnetic signature of their natal beach explains why relocation of nesting beaches is often unsuccessful. The magnetic coordinates are fixed by geology and planetary physics. You can't move them with the sand. It also explains why artificial lighting is such a serious threat to sea turtles. The first critical navigation challenge, finding the ocean, is disrupted by light pollution. If hatchlings crawl the wrong direction and exhaust themselves before reaching the water, they never get the chance to learn their natal beach's magnetic signature. The navigation system is disrupted before it can even begin to develop. Human activities that create magnetic anomalies, underwater cables, metal structures on or near nesting beaches, could potentially interfere with the imprinting process or with the homing ability of adult turtles. While the effects aren't fully understood, these are concerns that inform conservation strategies. The study of sea turtle navigation continues to reveal new details. Recent research has shown that even the tiny movements turtles make while still in the nest before hatching may play a role in magnetic imprinting. The turtles are sampling their magnetic environment before they even see the outside world, laying the foundation for the navigational abilities they'll need in the years ahead. As we reflect on the journey of a sea turtle, from hatchling to adult, from beach to open ocean and back, we see a beautiful example of how evolution has crafted navigation systems that are precisely tuned to an animal's ecological needs. Multiple cues, multiple strategies, developing and integrating over a lifetime to support one of nature's great migratory journeys. Integration and calibration, the symphony of navigation. Throughout this journey, we've encountered magnetic compasses, celestial stars, chemical trails, sound pictures, mathematical integration, visual memories. We've seen how different animals use these tools in different ways, adapted to their specific needs and environments. But we've also seen something else. These systems don't work in isolation. They interact, they calibrate one another, they integrate into unified spatial representations. This integration is what transforms a collection of individual sensors into a robust, flexible navigation system. Let's explore how this integration works and why it's so important. Consider a migratory bird preparing for its autumn journey. It's evening, and the bird is ready to depart. How does it determine which direction to fly? First, it might use the setting sun and the patterns of polarized light in the twilight sky to establish an initial heading. This sun compass gives it a directional reference. As darkness falls and stars become visible, the bird can identify the center of celestial rotation and use its star compass to confirm or adjust this heading. Simultaneously, it's sensing Earth's magnetic field, using this as another directional cue. Now here's the crucial part. These three systems, solar, stellar, and magnetic, all provide information about direction. In a perfect world, they'd all agree. But in reality, there are small discrepancies. The magnetic pole isn't quite at the geographic pole. There's magnetic declination that varies with location. The bird's internal clock might not be perfectly synchronized to local time if it's recently traveled across time zones. So, the bird's brain must integrate these cues, weighing each based on its reliability. On a clear night with stars visible, the star compass might get more weight. On a cloudy night, magnetic cues become more important. This isn't a simple averaging. It's a dynamic, context-dependent process that considers the quality and availability of each cue. Researchers have demonstrated this integration by creating conflicts between different navigation cues. When scientists use magnetic coils to artificially rotate the magnetic field around caged birds, the birds initially show confusion. Their magnetic sense is telling them one thing, while visual cues and other senses indicate something else. Over time, many birds show a compromise, orienting in an intermediate direction that averages the conflicting cues. This suggests that the navigation systems in the brain are communicating with each other, comparing information, identifying conflicts, and reaching a consensus direction. The neural architecture that implements this is still being investigated, but it likely involves regions of the brain that receive input from multiple sensory modalities and integrate them into a unified spatial representation. Calibration is another crucial aspect of navigation system integration. Calibration means using one reliable cue to adjust another cue that might be less reliable or that might drift over time. We saw this with the sun compass, which must be calibrated using an internal clock. The clock itself needs to be calibrated to the local light-dark cycle, which the animal does by observing sunrise and sunset. But calibration can happen between different navigation systems too. Young birds use celestial rotation to calibrate their magnetic compass. During a sensitive period before their first migration, they watch the night sky rotate around the celestial pole. This gives them a fixed, reliable reference for north. They then associate this learned north with the direction indicated by their magnetic sense. This calibrates the magnetic compass, establishing which magnetic field direction corresponds to geographic north. If you raise birds under an artificial sky that rotates around a different point, they calibrate their magnetic compass to this false north. When later released, they'll orient according to their calibrated magnetic compass, which now points in the wrong direction because it was calibrated against false celestial information. This elegant experiment demonstrates both that calibration occurs and that the star compass is treated as the primary reference for calibration. Why does calibration matter? Because sensory systems can drift or be influenced by local anomalies. The magnetic field has local variations due to geological features or human-made structures. An internal clock can become desynchronized. By regularly calibrating one system against another, more reliable system, the animal maintains accuracy. The integration of path integration with landmark information, which we touched on earlier, is another example of calibration. Path integration provides continuous positional updates, but accumulates errors. Landmarks provide accurate positional information, but only at specific locations. By using landmarks to reset the path integrator when they're encountered, animals combine the advantages of both systems. Desert ants demonstrate this beautifully. As an ant returns home using path integration, it's also watching for familiar visual landmarks. When it recognizes a landmark, it can update its estimate of position. If the landmark indicates the ant is closer to home than its path integrator suggests, the ant adjusts. This prevents accumulated path integration errors from leading the ant astray. Researchers have also discovered that different compass systems may operate at different spatial scales. For long-distance migration, birds might rely primarily on magnetic and celestial cues, which provide consistent information over large areas. For local navigation, within the familiar area around their territory, they might rely more on landmarks. This hierarchical organization, where different navigation strategies dominate at different scales, is computationally efficient. The sensory integration we see in animal navigation has inspired robotics researchers. Building robots that can navigate reliably in complex changing environments is challenging. By studying how animals integrate multiple sensors, filtering out noise, detecting conflicts between cues, and dynamically adjusting which sensors to trust, engineers have developed better algorithms for autonomous navigation. What's particularly impressive is that all this integration and calibration happens automatically, without conscious effort. A bird doesn't think, well, my magnetic compass says this, but my star compass says that, so I'll split the difference. It just orients in the appropriate direction. The integration is implemented in neural circuits, shaped by evolution and refined by learning during the animal's development. This brings us to an important point. Navigation systems develop. They're not fully formed at birth. Young animals go through critical periods where they learn to use different cues, where they calibrate their systems, where they build up memories of their environment. Disrupting these developmental processes can have lasting effects on navigational ability. For example, birds raised in environments where they can't see the sun may have difficulty using the sun compass later in life. Salmon raised in hatcheries under artificial conditions may not properly imprint on their natal stream's odor. Sea turtles that don't experience the magnetic field of their natal beach during a critical period may have impaired homing ability as adults. This developmental aspect has implications for conservation and animal management. Reintroduction programs must consider whether captive raised animals have had the opportunity to develop normal navigation abilities. Habitat modifications must consider whether they disrupt the sensory environment that animals need for navigation development. As we near the end of our journey through the world of animal navigation, take a moment to appreciate the sheer complexity and elegance of what we've explored. Single cells detecting magnetic fields at the quantum level, neural circuits implementing trigonometric calculations, learning systems that extract statistical regularities from environmental cues, multisensory integration that combines information across modalities. All of this machinery, evolved over millions of years, enables animals to solve one of life's fundamental challenges, finding their way through space. Closing reflections, a world beyond our senses. We've traveled far tonight, from quantum mechanics in a bird's eye to stride counters in an ant's leg, from star patterns wheeling overhead to chemical trails flowing through water. We've seen how animals, as different as bats and butterflies, salmon and sea turtles, have each evolved their own solutions to the navigation problem. What emerges from this exploration is a sense of the richness of sensory experience in the animal kingdom. So much of what guides animals through their world is invisible to us. We can't see Earth's magnetic field. We can't perceive the polarization of skylight. We can't detect the subtle chemical gradients that guide a salmon upstream. We can't hear the ultrasonic echoes that paint the world for a bat. Yet these senses are real. They're physical. They're measurable. The magnetic field is there, whether we perceive it or not. The polarization patterns exist in the sky. The chemicals float in the water. The echoes bounce back from objects. Animals have evolved to perceive and use this information because it helps them survive. This should give us humility. Our human experience of the world, rich as it is just one narrow slice of what's actually out there. The reality we perceive is filtered through our particular set of sensors. Other animals, with different senses, experience different realities, equally valid, equally real. It should also give us wonder. The fact that a tiny ant can navigate precisely across a featureless desert, that a bird no bigger than your hand can fly from Canada to Mexico and back, that a turtle can cross an ocean and return to a specific beach decades later. These are miracles of biology. They're mysteries that science has only partially unraveled. And they should inspire us to conservation. These navigation abilities depend on a sensory environment that humans are rapidly changing. Light pollution disrupts the natural light gradients that guide hatchling turtles. Noise pollution may interfere with acoustic communication. Chemical pollution alters the olfactory landscapes that animals navigate. Magnetic anomalies created by human infrastructure could affect species that rely on magnetic sensing. Understanding animal navigation isn't just an academic exercise. It informs how we design protected areas, how we manage wildlife, how we mitigate the impacts of human development. It reminds us that animals need more than just physical habitat. They need a sensory environment, a landscape of cues and signals that their navigation systems evolved to read. As you drift off to sleep tonight, think about the animals out there navigating right now. Birds flying overhead in the darkness, following magnetic field lines and star patterns. Moths detecting pheromones on the night breeze. Bats painting the air with sound. Salmon swimming upstream toward home. Turtles gliding through the sea. Reading magnetic maps written in the structure of the planet itself. They're all finding their way, guided by senses we barely understand, driven by instincts refined over millions of years. They're solving problems that would challenge our best engineers, using biological computers that run on milliwatts of power. They're navigating Earth. And in a small way, by understanding their journeys, we've navigated closer to understanding the beautiful complexity of life on this planet. If tonight's exploration sparked your curiosity, if these quiet wonders helped you unwind, consider liking this video and subscribing to Bedtime Biology. Share your thoughts in the comments. What navigation ability would you most want to experience? Which animal's journey intrigues you most? Until next time, sleep well, dream of distant horizons, and remember, you're part of a world far more remarkable than we usually pause to notice. Good night.